addition, you have also learned that they can have linkage, and the linkage is the linkage between the polypeptide chain is by the linkage occurs because of the peptide bonds between which is between the NH and CO group, and this kind of a peptide bond makes the protein structure which is a polypeptide. You have also learned that the when the peptide chain is formed, there is also SH group in some of the amino acid which can form the disulfide linkage. So you have also seen that the peptide bond and disulfide linkage, these are covalent bonds. So now you see in the primary structure of the protein, basically what you have is that it is peptide chain which is made up of mostly covalent bonds. Now, uh, after learning about this, we have also talked about that the proteins, they can have their structure, which is not only, I'm just going to share the PPT. So let us see how the protein looks like. Just a second. So first you have seen that the proteins, they look like a straight chain, and this straight chain of the protein, they are, because of the formation of peptide bond. Now today, what you are going to learn is about the secondary structure of the protein. So you understand what is the protein primary structure. In primary structure of protein, you have learned about the covalent bonding, which is occurring because of the presence of different amino acid, and the cross-linking is occurring because of the cysteine residue. And these cysteine residue, they form, cross-link, and that is what gives the interaction within the chain as well as between the different chains. Now we are moving on to another type of, protein structure, which is hierarchically, it is a higher structure. It is better than the primary structure in terms of their folding. So we are going to talk about the secondary structure of protein. So in the secondary structure of protein, polypeptide chain folds into regular repeating structure. Can you tell me what structure will be repeated? What is the common repeat in the protein backbone? So whenever protein is having amino acids, and these amino acids, all of them, they are linked together by peptide bonds. So what you have is CONH, which is in chemistry you call as an amide bond. We call it the peptide bond. So now in this bond, the thing which is repeated is CONH. Now this CONH, they can interact with each other by, what should be the interaction? It should be by hydrogen bonding. Now, because what is the donor here? H. Your hydrogen donor is NH, and hydrogen acceptor is a CO. So hydrogen bond can be formed between CO and H group, which is present in the peptide chain. So this is a repeating chain structure. What is non-repeating structure in a protein? Any one of you? So can you tell me what is a non-repeating structure in a protein? You can write your comment. Yes. So it is basically the R group, which is non-repeating structure. So proteins, the amino acid which are different, they are because of the R group. Now I have started sharing. Can you see the PowerPoint? The worst thing is that I can't hear you because of the some internet problem. Anyway, so polypeptide chains, they fold into regular repeating structure. It was Linus Pauling and Corey. They proposed two types of such periodic structure. So what are these periodic structure? First thing is very clear that these are occurring because of the hydrogen bonding. Now, second thing which is very important is this is occurring because of the neighboring groups. It is not that the first amino acid interacts with the 100th amino acid and gives the secondary structure. It is always that the one which is a neighboring good amino acid, they are interacting. So we'll talk about which amino acid can interact with which amino acid when there is a hydrogen bonding between CO and H. But one thing is very, very clear that these kind of a hydrogen bonding gives to different shape are different structures. These structures were recognized as alpha helical structure or alpha helix, beta pleated structure, that means a beta sheet structure, and other structures like beta turn, omega loop, etc. Now, coming to the structure, how it looks like, it's very important that you should understand how they look like and what is their, the actual configuration within the protein structure. So if you look at the hydrogen bonding, you see there is a backbone NH group which reacts with carboxylic group or carbonyl group, and that is the way the hydrogen bonds are formed. So this is the hydrogen bonding capacity within the amino acid chain, which is made up of peptide bonds. This alpha helix is tightly coiled backbone which forms inner part of the rod. So suppose I have a rod here. So I'm looking at the structure, which is the A structure. Here I have a rod. On this rod, what you will find is that there is a helix which is moving around this rod. So this is a solid structure. I will show you this. You have a solid rod-like structure around which there is a coil. Now looking at the coil, what you find is that this is folded on a backbone. So what is the backbone? The backbone is formed because of the main chain, and the side chain is nothing but R group. So these chains, they are folding around a rod, and they are they're in what they have is that their side chains, they are extended outwards. So if you look at the structure, A structure, the outward side is, what is the outward? This is basically your side chain. So these green color, you can see that the side chain, which is protruding out of the chain. So this is the main chain, which is a forming helix. And in this helix, R groups are present on outside. We will slowly discuss that what are the different types of model, and it is basically for your understanding. The one which I have given, which we are discussing right now, is known as a model which is called as a ribbon shape model. You can see a ribbon which is folded around the rod. The second one is the model of a protein which is called as a ball and stick model, and this is showing the hydrogen bonding. So you can see there is a hydrogen bond. I will tell you that these hydrogen bonds are not present in vicinity like first amino acid interacting with the second. It is a particular pattern which you will find. So that is what I will show you. And what you also see is that the stabilization of this helical structure is occurring because of the hydrogen bonding. So when there is a helix formed, unless something is holding this helix, the helix will not be stable helix. So what you have is that when there is an inner part, which is a rod, and this rod is a main chain where the side chain is extended on the outer verts, and they are stabilized by the hydrogen bond, and hydrogen bond is between carboxylic group and amino group of the amino acid, which is present as amino acid residues in the peptide chain. So where do you see that the amino acid is not interacting or not forming hydrogen bond? This happens at the terminal end. For example, this amino acid, the terminal one, which I'm showing you, is the one which will not form hydrogen bonding because it doesn't have a neighbor beyond its presence. Same way, this one, the last one in the chain, will not bond with the hydrogen bond. So ultimately what happens, there is a pattern which is going to appear, and this pattern is, okay. what you see is that I'm just ending the show to let you know. Okay, so what I wanted to see is that yes, it is a visible structure. So you have learned that there is a protein structure which can be given by a ribbon shaped model. There is another way of representing protein is a ball and stick model. And there is another way of a protein where you can give space filling model. You will learn that how these models are important. For example, this kind of a shape gives us a kind of a feeling that how much alpha helix or beta sheet is present in the protein structure. So it's a very clear indication. The space filling model gives us what type of amino acid is present in the structure. I will show you some of the slides to explain this better, where you will find that what is made up of interior core, what is made up of, what amino acids are making the exterior core of a protein structure. So that is what is, you can learn from the different models, and that is why different models have been proposed. Now, coming to the next part of it, if you look at the chain, I'm showing you a chain on the bottom in the figure, and this is nothing but primary structure of the protein. Now, start with the primary structure, and what you find is that, tell me what you are looking at this protein structure. This is the first amino, this is the chain of amino acid. Now, in this chain, you have NH. I'm starting with N terminal. So this is the N terminal of the amino acid. Let me explain it to you that this is the N terminal of amino acid. This N terminal, this is the C terminal, and that's why you see them free. And what you have, each carbon is having a side chain and one hydrogen atom. Each one you will find that one side chain and one hydrogen atom. So if I start with R1, which is a side chain, it will be R1, then R2, R3, R4, R5, and so on and so forth. Now, what you are looking at is that one of the side chain on this form side and another side chain is on this side. Why it is so? You have learned that there is a transform in which the amino acid is having the distribution of R group. So that is what you see, R group one on this side, another on this side, and this avoids the steric hindrance in the chain. Now, another very interesting thing which you will learn is that you have first carboxylic group here, and then this is the second one, this is the third one, and this is the fourth one. So what you have is that every, if I look at the amino acid, you will find that after, every third amino acid, that means fourth amino acid is a link with the first amino acid. This is the amino acid 1, 2, 3, 4. So in the fourth amino acid, the amino group is interacting with the first amino group, carboxylic group. Now, look at the second amino acid. The second amino acid, carboxylic group, is interacting with this amino group. So what you find is that regularly it is the fourth amino acid which is interacting with the first amino acid. And in whole chain, if I look at, what you will find is that there is a 3.6 amino acids. So it is averaging out to find out how many amino acid residues per turn they are interacting. You will find that there are 3.6 amino acid residues, because at the terminal they will be left out. They are not going to interact. So that is what is the way the alpha helix is formed. So looking at this, each residue is related to the next one by a rise of 1.5 angstrom. What it means is that if I have the first amino acid, this is the first amino acid, the next amino acid will be at a rise of 1.5 angstrom. So they are differentiating. So if I find out a distance between this carboxylic and this carboxylic group, it should be around 1.5 angstrom. And what will happen, and there is a rotation of 100 degrees. So if you look at this figure, this one, the helical structure, Now, if I look at this structure, what you see is that there is a helix form. Why helix is formed? Suppose you have one amino acid. When the second amino acid comes, there is a turn, there is an angle. And if there is an angle, what will happen? After a few angles, they will form a helix. So you find this kind of angle after each residue. So and along the helix axis, and this rotation is around 100 degree. And what you find is that this leads to the formation of alpha helix, which is normally 45 angstrom long. So what you have is around 4.5 nanometer long alpha helix you have. Now question is, do we have a protein which is just 4.5 nanometer long? Answer is no, because there are more than two or more helix, they can entwine, and by entwining what they can make is they can make a very stable structure which can have a length around 1000 angstrom. So if I look at the chain, these chain could be around 1000 angstrom, around 100 nanometer. Later on, you will learn that this does not mean that the protein is having a dimension of 100 nanometer, because these alpha helix structure will be further folded. That you will learn when we will talk about the tertiary structure. I am taking a alpha helix, opening it completely, and showing you what could be the length. But it does not mean that the protein itself will show this particular structure. So it is just evolution showing that how the primary structure folds to alpha helical structure. Now what you have is that such alpha helical coils are found in proteins like myosin, tropomyosin in muscle, fibrin in case of blood clot. It is also present as alpha keratin in hair. So these are present in many, many places. Now this helical cable in these proteins serve as a mechanical role in forming stiff bundles of fibers as porcupine quills. You know porcupine quills? They are like, they are their backbone, or it is like their tail, which is protruding out. So that is the way it looks like. The internal scaffolding or cytoskeleton of the cell is rich in so-called intermediate filaments, which are also standard a alpha helical coiled coils, and many proteins that span biological membrane, they also forms the alpha helix. What it means is that alpha helix is present in many, many forms. It is present in different types of proteins. And what you have seen so far is that the alpha helix can be present individually. Alpha helix can entwine with each other. They can form a long structure, and they can also, make coiled coils like you have seen here. So here I'm showing you largely a helical protein, which is present in the ferritin. Ferritin is a protein which is iron storage protein. You can see similar structure where two helix wind around one another to form a superhelix. So this is kind of a superhelix. So you have one helical structure which is running like this. There is one structure, and then you have another structure which interacts with the first structure and forms a kind of a structure which you can call as a superhelix. So do you understand what is a superhelix? Uh, I think, you understand? Okay, I'm going to show you the superhelix. That is where I was asking the question. The superhelix structure, I don't know why I'm not able to share the slide right now. Yeah. Can you see this structure? I'm going to, yeah. So this structure, which is the second part on the right hand side, there is one helix. Now this helix is interacting with another helix, and that is what is called as a super helix. So what they have happens is that they wind around one another, and that's the way the super helix is formed. Now moving on to the, so you have learned about the alpha helical structure. Now there are other structures which are also present, and these other structures are, one of them, very common one, is a beta sheet. So beta sheets are stabilized by hydrogen bonding, like you have learned that the secondary structure is having beta sheet structure, and they have the, the kind of interaction. So they have polypeptide chain. They will be called as a beta strand. I will show you what is the beta strand. And beta sheet is almost fully extended rather than being tightly coiled, as you have seen in alpha helix. So in alpha helix, what you have seen is that in alpha helix, you have looked at the structure, which is coiled structure. And this coiled structure is extended structure, whereas beta strand or beta sheet is fully extended rather than being tightly coiled. So there is no coiling in a beta sheet structure as compared to alpha helix. Adjacent in beta strand is approximately 3.5 angstrom. So you see the distance is also slightly more. The beta sheet is formed by linking two or more beta strand hydrogen bonds. So again, the kind of a bonding here is a hydrogen bond. So beta sheet and alpha helix both will qualify for understanding the secondary structure of the protein. So both alpha helix and beta sheet come under secondary structure. Now, if I look at the previous concept which I talked about, Ramchandran plot. Now, if I look at which kind of angle will give you the best stable structure of beta strand, here the red zone, which is showed here. So based on the angle which is present for this structure, will give you historically allowed conformation, or the one which can have the best stability. Okay. Now, looking at the beta sheet, first let me just show you this open beta strand. So beta strand, I'm just showing you one strand. It is not showing any hydrogen bonding at present. So slowly I will show you where the hydrogen bonding occurs. So what you have here is, these are, the green one is, okay, let me go back and show you the actual color. This is the side chain, and the center one is showing you the main chain. So there is the main chain of peptide, and you have the side chain. So what you find is that adjacent chain in beta sheet can run in opposite direction. So suppose I have a peptide chain here, you will have another peptide chain which is running here, and this peptide chain could be in parallel direction, anti-parallel direction, could be, in both ways, mixed one. So I'll show you that. So in anti-parallel arrangement, the NH group of carbon CO and NH and CO group of each amino acids are respectively hydrogen bonded to CO and NH of a partner adjacent chain. I'll explain it in the next slide. The parallel arrangement of each amino acid, the NH group is a hydrogen bonded to CO group of one amino acid on the adjacent strain, whereas the CO group in a is bonded to NH group on the two residue farther along the chain. So this part I'll just go to explain. Many strands typically have 4 to 5, but they can be as many as 10. What does it mean? That means I have shown you one strand. You can have 4 such strand, 5 such strand, and what they will do? They will run either parallel or anti-parallel, and all of them, they will have a hydrogen bonding. And other than hydrogen bonding, it is also possible that not only 4 or 5 strands they are interacting with each other. There may be 10 such strands which are interacting with each other. So you have extensive interaction within the beta chain. So it is very clear that beta sheet can be purely anti-parallel. So you can have just anti-parallel, you can have a purely parallel, or you can have both parallel as well as anti-parallel. Most of the time, fatty acid binding protein, this is a class of, this is a type of protein which binds to the fatty acid for lipid metabolism, are built of almost entirely beta sheet. So if I say exclusively where beta sheet is present, so it is the fatty acid binding proteins which show the, entirely made up of beta sheet. Okay, so in the next slide, it shows the different patterns of the beta sheet. Can you see the, pattern which is here in the first one? What you see is that there is a sheet which is running, and the direction is from here to here. So you have one running in one direction, another running in this direction. This is shown by these arrows. So there are two chains, and how we organize, how we say that the chain is running from one direction to another direction. Normally, N to C terminal, if I look at one N terminal, so one starts with N terminal, ends with C terminal, another starts with the, the other side, it will be C terminal and N terminal. So it's a anti-parallel. So these sheets are anti-parallel. They run in the opposite direction. The second one, this one, shows you that both of them are running in the same direction. Now, the difference between these two is the type of interaction they are having. You look at the interaction. This is the interaction which is occurring in case of anti-parallel, whereas this is the type of interaction occurring in the parallel. So in the parallel, how the interaction occurs between different amino acid of different chains is this one, whereas this is showing you the one which is anti-parallel. So anti-parallel and parallel, they show the difference in terms of hydrogen bonding between the different group, as I stated in the previous slide. Now you can have a mixed one where two of them are in the parallel direction and one of them is anti-parallel. So you can have different arrangement of the beta sheet. These are some of the, examples which will explain and which gives you idea what beta sheet is. So you see here, what we have is we have different beta sheet running in a protein structure. So here one beta sheet, another beta sheet. So one is running parallel, another is anti-parallel. So you have different types. So you see the same thing which has been shown in the A structure in the ball and stick model is shown in the, what is this model? Ribbon model. And ribbon model, what you see is that it shows direction. So these two are anti-parallel, these two are parallel, this is parallel, this is again parallel, and last one is showing anti-parallel. So these two are showing anti-parallel. So you have a different way in which these beta sheets, they can interact with each other. Now looking at this structure, now bottom, the C1, this one, what you look at, what is seen? It is a complete tertiary structure protein. It's not the secondary structure. What you have seen here is that you have alpha helical structure, this is alpha helix, and then you also see the beta sheet. Some of them, they are also showing beta turn. So I'll talk about what is this turn, what is the linkage between these. So these are kind of a beta turn. So I'll explain this in the next slide. So there are certain reverse turn or loops. They are also called as the beta turns. So most proteins have a compact globular shape. And because it is a globular shape, it cannot be extended alpha helix. It cannot be beta sheet. So what they have is that at different points, what happens that the globular shape requires reversal of a direction of their polypeptide chain. So suppose, let me try to explain. It's slightly, tricky unless you try imagining. So suppose a chain is going in this direction. What will happen at the terminal point? They will reverse back. They will form a kind of a loop. So there is a ith amino acid, I plus 1, I plus 2, I plus 3. And what will happen after taking this turn? There is a hydrogen bond. So because of this hydrogen bonding, it will keep a kind of a turn or loop-like structure. So these loop-like structures are formed in the protein structure. Many of these reversals are accompanied by a common structural element called as a reverse turn, or a beta turn or hairpin bend. So there are different names for this kind of a turn. In many reverse turn, CO group of a residue ith of the polypeptide is hydrogen bonded to NH group of I plus 3. So this is the kind of a pattern you see. So for suppose this is ith amino acid, it's a third amino acid which is a binding over here. And what are these bonds? This is a CO-NH bond. This interaction between this stabilizes the abrupt chain in the direction of the polypeptide chain. Suppose this is going on this direction, the polypeptide chain is going in this direction, there is abrupt chain, abrupt fold. Now this fold will not stay. This fold, unless there is a very long chain or unless there are many interactions, it will not hold together. So what you have is you have a hydrogen bonding, and this hydrogen bonding stabilizes the structure. And also the direction of the polypeptide chain. So you can have a globular shape. So shape is being maintained by this kind of a structure. Loops, they do not have a regular structure, a periodic structure, but often rigid and well defined. So you will find that the loop-like structure which is formed, it is a defined structure, it's a folded pattern, but it is not a regular pattern as you have seen in the beta sheet. What I mean to say here is that you see a pattern. This is a kind of anti-parallel pattern. This is again anti-parallel pattern. On the bottom, you are seeing couple of parallel pattern. So that kind of pattern doesn't happen, but at times they fold, they form a loop. So you can see there is a loop formation which is occurring. So loop, they don't have the regular pattern, but they are well defined. Turns and loop invariably lie on the surface of the protein. What it means is that if I have a protein structure, there is something which is in a core, there is something which is on the on the surface. So these loops, they are mostly present on the surface of the protein. Thus, if it is a present, what they will do, they will interact between the proteins and the other molecules. So they are the one which are providing interaction. So suppose there is interaction with the carbohydrate, these loops, they will be involved. The distribution of alpha helix, beta strand, and turns along a protein chain is often referred as a secondary structure. So this is the definition. But in general, if I say, what is the secondary structure? What is imagination? You can imagine that the structure which is formed because of the neighboring interaction of the peptide chain or neighboring interaction of CONH group, that is what leads to the secondary structure. And in turn, this interaction leads to alpha helix, this interaction leads to beta strand, as well as turns. Now then we move on to further complex structure, which is the tertiary structure of the protein. Most of the water-soluble protein fold into a compact structure, and when such kind of a structure is formed, I think you have to imagine what kind of structure they are forming. And let me just show you first. So this kind of structure is formed because of the complexity. I'll describe this further. So what you have is this is a complex structure. This is a kind of a shape you has made. It's a ball and stick model. And if I look at this other structure, this is the shape. In this shape, what you find? You find, though it is a mostly made up of alpha helix, but it can also have a beta sheet. It can have a turn. It can have, connecting links. So basically, anything which is formed of the secondary structure, but at the, at a global level, where the interaction is stabilized, not because of the neighboring amino acid, neighboring COH group can have interaction between the amino acid residue which are far apart from each other. So in this structure, for example, this amino acid can interact with this amino acid. So they are not neighbors, they are far apart from each other. Or this amino acid can interact with another amino acid which is present. So here interaction can occur in the global level. Whereas in the secondary structure, they will be in the neighboring amino acid. Now looking at one of the structure, let's first go to the definition and other details. The folding of most of the proteins is in a complex is divided of the symmetry. So there is no symmetry. You will not say that all of them is in beta sheet or in alpha helix. It may be made up of both alpha helical structure and so overall a course of the peptide chain formed in the protein because of the folding, they lead to the tertiary structure. So distribution of a polar and non-polar residues reveal the key facets in the protein or key structure. What does it mean? It means that the proteins which are making a kind of a complex structure, the distribution of a polar and non-polar amino acid. Non-polar amino acid like alanine, like valine, like leucine, and the polar amino acid like the one which is a charged one, like histidine, like your arginine, like your glutamic acid, all the charged ones, and the one which can have develop polarity on ionization, like OH containing groups, etc. So they are the ones which give the key structure of the protein. Now in aqueous environment, what is expected? Protein folds, and this is driven by strong tendency of a hydrophobic residue to be excluded from the water. This part we have discussed when we were talking about the non-covalent interaction. So what will happen if I take a protein structure which I defined as the primary structure, put them in the water? What will happen? The amino acids which are made up of hydrophilic amino acid or the polar amino acid, they will be on the surface. The one which is the hydrophobic, they will be going inside the core of the structure. I'll try to explain it. So the polypeptide chain folds so that the hydrophobic side chains are buried and its polar charge chains are on the surface. This is happening because it is in water. Suppose I take the same protein, put them in another organic solvent, the structure will be different. But in all the organism, they are present in the water. So when we are talking about the biological system or the system where we are talking about inside the cell, they are present in such a way that the hydrophobic side chains are buried and polar charge chains are on the surface. I will take a couple of example where this rule is not followed, but that we will take little later. Now, many of alpha helix and beta strands are amphipathic. Amphipathic means that they can have both hydrophobic phase as well as they can have hydrophilic phase. So they have beta strand has hydrophobic phase, which points into the protein interior. So if we have two structure in a strand, they will the hydrophobic part will go on the inner part, and the polar part will go out of the outer side of the structure. Unpaired peptide NHCO group markedly prefers water in non-polar milieu. Why it is so? Because the NH group and CO group can have hydrogen bonding, and that's why they will prefer water. They will not go. So if NH and CO, they are not interacting with each other, where hydrogen bonding is not present, they will prefer hydrogen bonding with the water which is present in the environment. Now here, the hydrophobic environment is giving you or giving the protein a environment where all the groups which are hydrophobic in nature, they are embedded. This pairing is neatly accomplished in alpha helix or beta sheet. So that's why your NH and CO group, they are interacting with each other. You have seen both in alpha helix and beta sheet, they're interacting. The van der Waals interaction is occurring in the hydrocarbon side chain. Now, within the beta sheet and within the alpha helix, you have seen that they are having hydrogen bond. But among these sheets, suppose I have alpha helical structure, which is present separately, beta sheet is present separately, they can also have ionic interaction. But whatever is going in the core of the protein structure, they will be having hydrophobic nature. There is a set of the amino acid. This I have already discussed. They can have different types of interaction. So this is the structure which I have already referred as a structure, which is the ball and stick model, where you have seen how they are present. This is another structure, the protein of myoglobin. Now you look at the structure. Myoglobin is an oxygen carrying muscle protein. It is a single polypeptide, contains 153 amino acids, and they also bind with oxygen. So where do they bind with oxygen? They have a central atom, which is protoporphyrin, is a heme group. So this is the heme group. Can you see here in the center there is a purple color ball which is present. This is the heme group. This is the group which is this shows this structure which is known as a schematic model of the myoglobin structure. It presents the heme structure and the iron atom. So this purple is a iron atom. It is surrounded by different parts of the heme structure. Now let us move on to the next slide, where it shows you a very interesting point. You look at A. A is showing a space filling model. Now this space filling model, if I say that yellow color amino acids, they are hydrophobic amino acids. So you can see some of the amino acids are hydrophobic, some of them of the hydrophilic. So in the three-dimensional structure, the amino acid which is yellow color, they are very less. But if I take the protein and cut it, what you will find is that most of them, they become yellow color. So this demonstrates, the cross-sectional view demonstrates that in the core, in the center, most of the amino acids are hydrophobic, whereas in the outer surface, many of them, they are hydrophilic in nature. So this demonstrates how the structure occurs. This is one of the example, and which is exceptional example. Now, there are proteins which are present in the membrane, and one of them is a porine. Now, this protein will have the hydrophobic amino acid, which is present on the outer surface. Why? Because these proteins, they interact with the lipids, and because they are mostly interacting with the lipids, their environment is hydrophobic, and that's why the amino acid which is present on the outer surface, they are hydrophobic, whereas the water is making a channel on inside, and that is why the inner core is the one which is hydrophilic in nature. This is another example which is showing the domain, the concept of domain. The compact globular unit called domains, and they range in a size from 30 to 400 amino acids. So some of them, they are functional unit of the protein, and they are the one which are making the domain of the protein. So you will have the domain, even if the tertiary structure is different, the domain may be similar between two different proteins. So this gives you a very brief outline of the secondary and tertiary structure. I have not given you much detail about it, but if you have any question, you can ask. You can have one or two quick questions. So have you understood about the alpha helix, beta sheet? Somehow we don't have any communication. I don't know why, but I cannot hear you, but probably you can hear me. Okay, so I'll end my class today, and I will reschedule yesterday's class in the next next Tuesday, so that we complete the protein structure. So in the protein structure, we have talked about the tertiary structure. Now we will be moving on to quaternary structure and few more details, and then we will cover the protein structure. Okay, then I will signing off. I'm going to sign off now. Any questions so far?